All right, so if you haven't already, please pull out your notes. We're going to finish them up. This is going to give you the advanced placement biology details for protein synthesis. So as we learned last week, the flow of genetic information goes from a gene to a protein. So in your space provided, please draw out the flow. It starts with DNA. After DNA, draw your arrow and make RNA. And then after RNA, draw your arrow and write down protein. The process of going from DNA to RNA is called transcription. The process of going from RNA to protein is called translation. All right, so let's go a little more detail into transcription. First of all, let's make sure we know that transcription goes from a DNA nucleic acid language to an RNA nucleic acid language. All right, so as you know, there are two sides of a DNA strand. One side is going to be transcribed into the mRNA and the other strand will not. So when we are making the mRNA, the transcribed DNA strand is going to be called the template strand. You can label that on your diagram right here. And then the DNA strand that is not transcribed into mRNA is called the coding strand. And that is the strand that's on the top, this one right here. That is a coding strand. We are going to make mRNA from our template strand, which is the one on the bottom. All right. Notice that the coding strand will have the same sequence as the RNA, except there will be the difference of T's and U's. So all of this happens in a bubble, as you can see in the pink. And you can see that the RNA is being made in green here. And that is already labeled for you. And the enzyme that is involved, please label that in your box, is called Rna Polymerase. It is similar to DNA polymerase, which did the replication of DNA. All right, there are actually 3 RNA polymerases that are involved with transcription. RNA polymerase number 1 is going to transcribe the genes to make rRNA. And so this enzyme is responsible for making the ribosome. RNA polymerase 2 is the main RNA polymerase when we think of an RNA polymerase, because this is the one that's going to transcribe the genes into mRNA, which is our messenger RNA. And then RNA polymerase 3 only transcribes the tRNA genes. Each of them have a specific promoter, remember, introductory beginner sequence that it recognizes to do its job. All right, so how does RNA polymerase know what gene and what part of DNA to read to make the mRNA? There is a promoter region. This is a binding site that's actually before the gene. The gene is in the blue here, and the promoter is before that. If you see on the promoter, there is a sequence called the TATA box, T-A-T-A, and actually that is where RNA polymerase will bind to, and that's when transcription would start. There are actually 2 binding sites. One is an enhancer region. That's a binding site for activators, which will activate the genes and turn them on. And there's also a silent region, which is a binding site for repressors, which would turn genes off. So one thing I want to add is notice on your TATA box description, it's a binding site for RNA polymerase, and it's the binding site for transcription factors. So let's go into what those are. So transcription factors are actually just a bunch of proteins that are going to bind to the promoter region on the DNA. You can see them on the picture. They're already labeled for you. They're called transcription factors. And so there are a bunch of proteins. You can see them in purple, like they look like little bubbles, and they actually bind to the DNA. And that triggers, once that binds, then it triggers the binding of the enzyme, the RNA polymerase, to the DNA. So the transcription factors bind first, and then the RNA polymerases can bind to the DNA. So then once RNA polymerase is bound to the DNA, then obviously RNA polymerase will do its job, and it will match the RNA bases, the opposite complementary letter, from the DNA to make the mRNA. So one thing that you might not have known is that eukaryotic genes have junk in them. They are not all pieces of DNA that actually code for genes. So they're not continuous. So if you look on the picture, you see exons and introns. Exons are the real gene. These are the ones that are going to be expressed and make a gene that code for the DNA. And then you can see in orange that you have introns in between those real genes, and that is the junk, and these are the in-between sequence. And so what we need to do is actually get those introns out. All right, so after mRNA is made, mRNA has to be edited. It has to be spliced. And so there is post-transcriptional processing that takes place. What happens is we have the pre-mRNA is going to go, it's our primary transcript. That's going to go through mRNA splicing. And so what mRNA splicing is basically we're just going to get those introns, that junk that doesn't code for any genes, out, and we're going to edit that out. Once those introns are out, then we have a mature mRNA transcript. So you can see in the picture of our DNA right here, we have our exons and our introns, and the exons are what we want, the gray part, and the introns are the junk that we don't want. We want to take those out. So here's our primary mRNA transcript. We're actually going to splice those introns out, and we are left with a mature mRNA transcript. And you can see that all the good genes, or the good code, is there. When we are splicing the mRNA, we have to be accurate. There is no room for mistakes, because if you just miss one letter, one single base, if it's added or lost, it's going to throw off the reading frame. So you can see in our DNA, we made our mRNA. And if just one letter is different, if this C, it looks like if this C got changed to a G, that turns and changes the amino acid sequence at the bottom. So we need to be accurate. So how does the splicing work? There are special enzymes, of course, for splicing the mRNA, and they're called SNRPs, SNRNPs, and they're just a small nuclear RNA. And so a whole bunch of them, you can see them in the picture right here. There's several of them. They'll join together and form a spliceosome. So a spliceosome is several of these SNRPs, and what they do is they recognize where the introns are, and they'll bind to it, they'll cut it, and then they'll paste the gene back together. So you can see that here, that you have a bunch of these SNRPs, the spliceosome, coming together and trying to get that orange intron cut out. And at the end of the day, we have our mature mRNA, and our intron is not there. So beside the splicing that has to happen on the mRNA, we also have other post-transcriptional processing that has to take place. The cytoplasm has enzymes in it that will attack mRNA. So we need to protect mRNA on its trip from the nucleus to the cytoplasm. So how are we going to do that? We're going to do two different things. To protect the ends of the molecule, we're going to add a 5', that should be a prime, Gtp Cap. It is a chemically modified molecule of Gtp. You've seen Gtp in photosynthesis during the cycle, the Calvin cycle. And so what the Gtp Cap does at the 5' end of mRNA, it facilitates the binding of mRNA to the ribosome. So it helps that RNA bind to the ribosome, and it protects the mRNA from being digested by these enzymes that are in the cytoplasm. They're called ribonucleases. And ribonucleases are enzymes in the cytoplasm that will basically break down and digest RNA if it's not protected by this Gtp Cap. So when we continue our lab, once you make your mRNA strand off of your DNA strand that should be done already, you are going to have to add a Gtp Cap on your 5' end. So you'll have to find your 5' end and then add a Gtp Cap piece on there, which I don't believe you have yet. Another thing that also needs to be done is we're going to add a 3' poly A tail. We're basically going to add 100 to 300 adenine nucleotides at the 3' end. So the 5' end gets the Gtp Cap, the 3' end gets a whole bunch of A's, a whole bunch of adenines. And what these adenines do, you can see them right here, they assist in the export of the mRNA from the nucleus to the cytoplasm, and they help the mRNA just stay stable. So you can see in the picture, please label, you have your 5' end here, your 3' end here, and at the 5' end we're adding the Gtp Cap, and at the 3' end we're adding a bunch of A's. All right, so moving on to translation. Translation is the language of RNA going to the amino acid language. So here's our genetic code, as you have seen and you know how to use. This code is for all living organisms. It is the strongest support with the protein synthesis that there is a common origin for all living things. It is redundant, which means there are several codons that code for the same amino acid. So for example, proline, right here in the middle, has 4 different combinations that will code for proline. So they call that a third base wobble. So CCU, CCC, CCA, CCG. That third base can wobble, and that doesn't even actually matter for in this case that it's proline. The start codon is always AUG, which codes for methionine, and the stop codons are always going to be UGA, UAA, or UAG. All right, so looking at the tRNA a little bit closely, it does have a clover leaf structure. The anticodon, which are the complementary letters that go against, or the complementary to the mRNA, is on the one end, and the amino acid is on the other end at the 3' end, actually. All right, so our tRNAs have an amino acid attached to it. There's an enzyme that bonds that amino acid to the tRNA. It's called Aminoacyl-Trna Synthetase, and so that's the enzyme that bonds the amino acid to the tRNA. This bond requires energy in the form of ATP. The bond that forms between the amino acid and the tRNA is very unstable, so that's why the amino acid can release easily when it's needed to at the ribosome when we're making our protein. So for the translation part for building a polypeptide, there is basically 3 parts. We kind of already did this before. Initiation brings together the mRNA binding to the ribosome subunits, and then we have a tRNA coming in at the P site, or the T site, and then it elongates, right? It moves to the A site and the P site, and we keep elongating, adding amino acids as we go based on the codon sequence. And then eventually we will reach a stop codon, and that will terminate the whole procedure. So the protein that gets made can basically be all kinds of different proteins, right? And so there's different destinations that that protein will go to once it's made. It can be secreted out of the cell. It can go to the nucleus. It can go to the mitochondria, the chloroplast, cell membrane, cytoplasm, etc. There's so many different areas because there's so many different kinds of proteins. For example, a protein can be a signaling protein, and maybe you need to go to the cell membrane to do that. So basically this picture shows you the whole picture of what we talked about, and basically it asks you and challenges you, can you tell the story? Can you tell a story if you are starting with a DNA molecule? How does a DNA molecule make the RNA? What kind of post-transcriptional processes have to occur to go from a pre-mRNA to a mature mRNA? And then once you have the mRNA, how do we actually make the protein? And so all these different terms are things that you need to understand to be able to tell the story. All right, so one last thing is what happens in bacteria? Like, how do they make their proteins? Because bacteria can make proteins too, but it's not as complex as a eukaryote cell. Because bacteria, prokaryotes, they don't have a nucleus, right? So their DNA is just one big circle, right? One big piece that's in a circle, and it's not linear like ours. And it can go through transcription and make mRNA, okay? But just remember, there's no nucleus in a prokaryote. All right, so filling out the chart, if we start on the left for the prokaryotes to describe the difference between prokaryote and eukaryote genes. For prokaryotes, our DNA is in the cytoplasm. It is not in the nucleus. There is no nuclear membrane. Another difference is the chromosome shape. Prokaryotes' chromosomes are circular. They're not linear like in a eukaryote. In prokaryotes, the DNA is naked. There is no protection around it. There's no nuclear membrane around it. It's just naked DNA sitting there in the cytoplasm. And then another difference that we haven't mentioned yet is that the prokaryotes don't have any introns to worry about. So there is no post-transcriptional processing, which has to happen for a eukaryote. All right, and so on your right, for the eukaryote column, You're going to put DNA is in the nucleus, right, with the nuclear membrane around it. The chromosome shapes are linear, right? We have X-shaped linear chromosomes. They're not circular like in a bacteria cell. And our DNA is not naked. It's wrapped around these histone proteins that allow it to not get tangled up. And eukaryotes have introns that we have to splice out and exons. They have both. So just a little bit of detail for how does translation happen in a prokaryote. Transcription and translation are happening basically at the same time, simultaneous, in bacteria. Because the DNA is in the cytoplasm, we don't have to worry about the nucleus and doing any of that. There is no mRNA editing because they don't have any introns. And the ribosomes are reading the mRNA as it's being transcribed. So everything happens a lot faster. So the difference between a prokaryote and eukaryote for translation is time. It's a lot faster. And there's a physical separation because we have a nucleus in the eukaryote and we don't have a nucleus in the prokaryote. It takes a eukaryote around 1 hour to go from the DNA to a protein. And that finishes up our notes.